Hey everyone, and welcome to this explainer. So, have you ever wondered what's actually going through a doctor's mind when they write you a prescription for antibiotics? I mean, from the outside looking in, it kind of looks like they're just picking a random medicine off a shelf, right? But the reality, it's actually fascinatingly different. Today, we're going to completely demystify this whole process. We're going to see how picking the right antibiotic is literally a highly logical detective process. It's this systematic framework where we match a suspected pathogen with a drug's specific spectrum of activity and the exact site of the infection. It's definitely not a guessing game, it is an absolute science of precision. To help us really wrap our heads around this, we've broken the whole clinical selection process down into a simple 5-step framework. We're going to look at 1. The prescribing funnel, 2. Profiling the bacterial suspect, 3. Targeting specific defenses, 4. Testing and de-escalation, and finally, 5. The patient factor. Let's jump right in. Section 1. The prescribing funnel. Let's arrive at the scene of the infection and get this first phase of clinical detection started. Okay, so a doctor's very first move is the clinical diagnosis. Essentially, they need to locate the crime scene. Is the infection in the lungs? Is it in the urinary tract? Maybe the skin? You see, different sites strongly suggest different usual suspects. For instance, community-acquired pneumonia almost always points the finger at a bug called Streptococcus pneumonia. After pinpointing the location, the doctor has to assess the severity. Like, is the patient stable or are they septic and need immediate, heavy-hitting intervention? Once we know the site and how bad it is, we move straight to step 2, initiating empirical therapy. Now, the concept behind empirical therapy, what we call the best guess, is super interesting. Think about it. When a patient is severely sick, doctors just can't wait days for a lab to culture the exact bacteria. They have to act fast. So empirical therapy is basically about casting a really wide net. It means starting the patient on a broad-spectrum antibiotic based on local medical guidelines to cover the most likely bugs that could be causing the problem. Really, it's all about holding the line. We want to stabilize the patient while we wait for that hard, microscopic evidence to finally come back from the lab. Section 2. Profiling the bacterial suspect. While we're waiting on those definitive lab results, it's time to zoom in and start mapping out the biological targets of our bacterial culprits. So selecting the right antibiotic ultimately comes down to matching the drug's specific pharmacological class to the specific bug. And the very first profiling decision we make here is based on the bacteria's physical armor, its cell wall structure. We actually figured this out using a super rapid first look test called gram staining. This test brilliantly illustrates the biggest divide in the entire bacterial kingdom. On one side, you've got your gram-positive bacteria, like staph and strep. These guys have this really thick, porous outer layer called peptidoglycan, and they hold on to a Perkel stain. Now, on the flip side, we have gram-negative bacteria like E. coli or Pseudomonas. These are trickier. They have a much more complex, highly protective double membrane structure, and they stain pink. Honestly, knowing this simple color-coded armor type literally dictates our entire prescribing strategy. Because once we know the wall structure, we can systematically match our weapons to it. For gram-positive bugs with those thick walls, we're going to deploy bactericidal weapons like penicillins or first-generation cephalosporins. Why? Because these drugs specifically target and literally smash that thick peptidoglycan layer, which makes them absolutely perfect for things like skin infections or strep throat. But let's say we're dealing with gram-negative rods hiding behind that complex double membrane. These are often the culprits in things like UTIs or sepsis. For those, we need drugs that have a higher affinity to penetrate that specific barrier, drugs like aminoglycosides or third-generation cephalosporins. Okay, let's move on to our next clue, which is all about the environment. Basically, where is our suspect hiding? More specifically, do they thrive in oxygen-poor environments? You see, some bacteria, which we call aerobes, absolutely need oxygen to survive. But others, known as anaerobes, actually thrive in the dark, oxygen-deprived corners of the body. We're talking deep intra-abdominal tissue, the gut, or even dental abscesses. For these specialized hiders, standard drugs just aren't going to cut it. They often fail completely. So we have to deploy specialized tactics. We'll use a drug like metronidazole, which is super cool because it actually activates inside the anaerobic cell to just shatter its bacterial DNA. Or we might use clindamycin, which works by completely blocking the bacteria's ability to synthesize proteins. Section 3, targeting specific defenses. So what happens when standard weapons fail against bugs that simply refuse to play by the rules? You know, the ones that lack normal cell walls entirely, or maybe they've developed intense resistance over time. If we map out how we escalate our force against these specialized bacterial defenses, it reveals this really fascinating pattern. Take atypical pathogens, like mycoplasma, which causes walking pneumonia. They literally don't even have a cell wall, so throwing penicillin at them, totally useless. Instead, we use macrolides. These drugs actually cross the human cell membranes and target the bacteria's internal machinery. Now, moving down the list, what if we have a high-risk MRSA infection? This is a bug that has actually modified its own wall to resist standard drugs. In that case, we escalate to vancomycin, which is smart because it binds to a completely different, unmodified part of that cell wall. And finally, for those terrifying multi-drug-resistant superbugs you find in the ICU, we have to bring out the heavy artillery, carbapenems. These are the broadest spectrum drugs we have, and they are structurally stable enough to withstand the destructive enzymes that these superbugs try to throw at them.
But here's the thing. A massive, broad-spectrum hammer isn't always the right answer. Sometimes the most elegant solution is actually just a little bit of sabotage. For specific recurring conditions, like your standard uncomplicated UTI, we can use drugs like Bactrim. So instead of trying to violently smash the cell wall, Bactrim sneaks in and targets the bacteria's metabolism. It literally starves the bacteria by blocking its ability to synthesize folic acid, something it desperately needs to grow. It's just a highly targeted, highly effective metabolic interruption.
Section 4. Testing and de-escalation. All right, our empirical phase is officially over. Those lab results from the cultures we took on day 1, they've finally returned, which means we can pivot from casting that broad net to using a highly precise sniper rifle. So the lab has grown the bacteria and run what we call antimicrobial susceptibility testing, or AST for short. Basically, they expose the bacteria to a bunch of different drugs and hand us back a report card. S stands for susceptible. That's our green light. It means the drug is likely going to work at a standard dose. Then we have I for intermediate. This means it might work, but we're going to have to pump up the dose. And R is resistant. That's a hard red light. Avoid that drug entirely. It won't work. The lab also gives us the MIC, which stands for minimum inhibitory concentration. This is basically the absolute lowest concentration of the drug you need to prevent visible bacterial growth. The lower the MIC number, the more highly sensitive the bug is to that specific weapon. Getting this report card initiates the single most critical pivot in our whole systematic framework, moving to definitive therapy. This is all about de-escalating our pharmacological force. What we do is look at that susceptible list, and we intentionally switch away from our initial broad-spectrum best-guess drug. We want to pivot to the absolute narrowest spectrum antibiotic possible, the one that perfectly matches the bug's specific vulnerability. And I really want to pause here for a second, because it is absolutely crucial that we de-escalate. You might be wondering, hey, why not just keep them on the big guns to be safe? Well, as this quote highlights, narrowing the spectrum directly protects the patient. It reduces the collateral damage to all their good, healthy bacteria, which minimizes nasty side effects. It also saves healthcare costs. But honestly, most importantly, it prevents the development of further antibiotic resistance out there in the broader population. Honestly, it's just responsible medical stewardship. Section 5. The patient factor. Okay, so we found our perfect biological match. But we aren't quite done yet. We still have to make sure that this perfectly matched weapon actually suits the unique human being sitting right in front of us. This is where we fine-tune our choice based on the patient's specific biology. First up, organ function. We have to ask, can their kidneys and liver actually handle clearing this drug? A lot of times we have to adjust the dosage based on a patient's kidney function or creatinine clearance, just to prevent toxicity. Second is allergies. Look, the absolute perfect drug on paper is completely useless if the patient has a severe allergy to it, right? If they're allergic to penicillin, we have to immediately pivot to a totally different chemical class. And third, and I find this one so fascinating, is site penetration. The drug has to physically be able to reach the infection. For example, if a patient has meningitis, well, many standard antibiotics literally cannot cross the blood-brain barrier. So we have to intentionally select a drug with a molecular structure that allows it to breach that specific anatomical fortress. As we wrap up this explainer, I really want to leave you with this final thought. Choosing the right antibiotic is definitely not some quick reflex. It's this rigorous, highly systematic process of matching a suspected pathogen with a drug's spectrum of activity and the infection site, all while balancing the patient's unique needs. Think about it. Applying this precise, logic-driven framework doesn't just save the individual patient lying in the hospital bed today, By intentionally de-escalating and surgically targeting our therapies, this exact framework is literally our primary defense against the creation of unstoppable, multi-drug resistant superbugs tomorrow. So, what does this mean for the future of global medicine? Well, it means this kind of precision medicine isn't just nice to have, it's absolutely vital for our survival. Thanks so much for exploring this with me today, and I'll see you next time.